All right, welcome everyone. Thanks for being here. Before I begin, so now the seminar series, you do get CME credit, so please sign in. If you're zooming in, then Pen and Fatuma can check who's zooming in, and they will follow up by email to see if you'd like to receive CME credit. And then last but not least, if you've missed past lectures and you're interested to watch those, all of these are being recorded, and now we have a YouTube channel so that you can click and watch videos. So you guys will be on YouTube soon. So today we feature two investigators from the Division of Pediatric Critical Care, Rebecca and Sanjeev. And so first they'll tag team. Rebecca will first present, and then Sanjeev. Just as a way of background, Rebecca was a medical student at Boston University, and then she was a pediatrics resident at Boston Children's, and then she came here to do PICU fellowship, and then did advanced clinical training as a fellow at Stanford, and then came back to us as junior faculty and is currently an assistant professor. She has been funded previously on NIH training grants and is currently funded on an AHA award. Sanjeev was MSTP at University of Washington, where he was studying fly genetics, and then came here as a pediatrics resident and also as a PICU fellow, and continued as a postdoc and a trainee in CVRI. Both of them continued as postdocs in Jeff Feynman's lab. And currently, Sanjeev is an associate professor, and he's been funded by the NIH, currently in R01, and in the past an AHA award. So today, Rebecca is going to talk about her work and trying to understand what happens in the context of RV strain, in the context of pulmonary hypertension, and Sanjeev, in a related topic, will discuss his work on trying to understand pulmonary lymphatics in the context of pulmonary hypertension. So please welcome me in joining Rebecca.
Thanks for the introduction, and thank you to the committee for the opportunity to speak today about my research. As was mentioned, I study the right ventricle and a system of congenital heart disease that I'll explain in pulmonary hypertension. And so I think everyone in this audience is familiar with right ventricular sort of normal physiology and then pathophysiology in the setting of increased RV afterload. So typically the RV is a crescent-shaped ventricle under low pressures, but in the setting of pulmonary hypertension, it dilates and impairs LV filling, and it also has increased oxygen demand coupled with impaired coronary perfusion pressure. And so the right ventricle is really kind of perfectly adapted under physiologic circumstances to the load it's meant to carry. These pressure volume loops, which I'll show a lot more of later, show that the area represented by the loops is the work done by those ventricles. So to pump the same blood volume, the RV actually uses about a fifth of the work of the left ventricle. And it can do that because it's pumping to this high compliance, low resistance circuit of the pulmonary vasculature. However, the RV classically does not do well when it's subjected to acute increases in afterload. And this is classic data from Brunwald that shows even with modest increases from 10 to 30 torr, the RV stroke volume goes down dramatically. And that's, of course, not true, as we know, for the left ventricle. And typically that's also the case with patients with pulmonary hypertension. We know that idiopathic pulmonary hypertension is typically not well tolerated by the right ventricle. Our patients suffer from decreased systolic cardiac performance, even in the setting of mild increases. And sorry, I get very breathless when I give talks when I'm pregnant, so if I'm panting, don't be alarmed. Anyway, just even in the setting of modest increases in pulmonary pressure, patients, typically the RV has depressed systolic output. And then you can see in these gross pathologic specimens, the kind of typical picture is the one at top where the RV dilates and thins in response to these increases in pressure. However, that's not the case for every right ventricle, and these are even idiopathic pulmonary hypertension patients. So the specimen at the bottom is a woman who for decades had pulmonary hypertension and actually had advanced lesions with the classic plexiform findings. But she had preserved systolic RV function. She actually died from colon cancer. And that's also true when we look at pressure volume loops and kind of invasive measures of cardiac performance. We know that some patients have preserved systolic function despite the same elevations in pulmonary pressure. So what makes these patients different? Well, one model that we can kind of look to is patients with Eisenmenger syndrome, which I'll explain more in a second, or really patients with pulmonary hypertension associated with congenital heart disease. And we know from decades-old survival curves that these patients tend to survive longer than do their counterparts with idiopathic pulmonary hypertension. And that's particularly true when you break these patients apart, as you can see on the right graph, to those with post-tricuspid lesions as opposed to pre-tricuspid lesions. And I'll talk about why that might be. A brief review for the trainees in the audience about what Eisenmenger syndrome is. It begins with a left to right shunt. And that leads to increased pulmonary blood flow. And this is a model that we take advantage of in our animal model as well. This early on leads to endothelial dysfunction and vascular remodeling, both in the pulmonary vascular endothelium, but also in the lymphatic endothelium, which Sanjeev will discuss, and ultimately an increase in PVR, which leads to an inverted shunt and cyanosis, which is really the hallmark of the Eisenmenger syndrome. And so I mentioned that these patients survive longer than do idiopaths, but they do it despite having advanced pulmonary vascular disease. When you measure that by pulmonary artery pressure and pulmonary vascular resistance, they're equally elevated in these patients. However, when you look at indices of RV performance, their right atrial pressures are lower and their cardiac indices are slightly higher. And so it could be simply that these patients have a pop-off, and that's certainly probably true to some extent. And these are things that Dr. Moore and some of our surgeons may be doing as palliative procedures for some of our patients with advanced pulmonary hypertension, creating a pop-off in these patients, either an ASD or a POTS shunt, and kind of in Wood's classic article where he initially described the Eisenmenger syndrome in detail, he said closing these defects is actually responsible for the most death in these type of patients because they rely on that pop-off. However, there may also be something fundamentally different about the right ventricle in these patients, and that's certainly our hypothesis in the lab that contributes to their improved survival. And there's some evidence to support that. So this is an echo study, obviously. So the patients on the left side are idiopaths and those with chronic thromboembolic pulmonary hypertension or CTEPH, and then Eisenmenger syndrome patients compared to controls who are in the rightmost panel. And when you look at echo indices of RV function, which are imperfect as we know, both in systolic PA pressure, TAPSE, RV fractional area change, and RV strain. And all of these indices, the Eisenmenger's patients are closest to the control population, except for the systolic pressure, PA pressure, compared to the other groups of PH patients. So again, a sign that not only do they survive longer, but it seems to be that they have superior RV performance. One reason why this may be true is that these patients have a preserved fetal phenotype. So as all of you know, in fetal life, the right ventricle is the dominant ventricle. It's responsible for 58% of cardiac output. It pumps against equal afterload, as does the fetal LV. And if you look at this is a fetal echo in the upper left panel, the ventricles are concentrically hypertrophied and have the kind of typical fetal pattern of a septum that is midline and concentric RV hypertrophy. The remainder of the panels are cardiac imaging obtained from patients with Eisenmenger syndrome of varying ages, and the oldest patient is actually 74 at the bottom right. And you can see the preservation, at least from a gross imaging perspective, of this fetal relationship, where they have concentric hypertrophy and a midline septum. And contrast this with the images at bottom of a patient who has RV failure in the setting of pulmonary hypertension. You can see the large dilated RV, the septum that's bowing into the LV and impairing LV filling. And so there's clearly physiologically a fundamental difference between these patients. And so kind of using this group of patients in our animal model as a jumping off point, if we could kind of choose the things that we wanted describe an adaptive RV. It would include preserved systolic function in the face of increased afterload, normal ventricular-ventricular interactions, increased angiogenesis, or at least preserved angiogenesis, and a lack of fibrotic response in the setting of hypertrophy. And one kind of paradigm that I'm working with in my research is that a lot of the research in pulmonary hypertension and in RV failure focuses a lot on initial compensated hypertrophy that sort of inexorably declines into right ventricular failure. And that's kind of the typical model when you look at animal models, for example, monocrotaline or pulmonary artery banding models. However, I think this is fundamentally different from the right ventricle and congenital heart disease. As I said earlier, the RV in fetal life is the dominant ventricle, and preservation of this fetal phenotype due to the hemodynamic forces, particularly of a post-tricuspid shunt, I think account for fundamental differences in the RV that persist and lead to more adaptive responses to increased afterload than does the RV that's remodeled in postnatal life and then is confronted in juvenile or adult life with increased RV afterload. And so our model that we use is fetal surgery. This model was actually developed by Dr. Reddy while he was here in the 90s, along with Dr. Feynman. So we do an aortopulmonary shunt. It's a large unrestricted shunt implanted in late gestation fetal lambs, about 2 weeks before spontaneous delivery. So this shunt is important because it doesn't really change fetal hemodynamics at all. It really just recapitulates the ductus. But what it does do is after birth, from the moment of that perinatal transition that we're all familiar with as pulmonary vascular resistance falls, that shunt is in place. And so pulmonary blood flow goes up, the fall in pulmonary vascular resistance is attenuated, and it more clearly mimics the changes in the pulmonary vasculature, and for my purposes also the RV, since it's an unrestrictive shunt, that occur in congenital heart disease compared to postnatal models of congenital heart disease where some of these postnatal changes have already happened because they're so rapid. And so we've looked at the right ventricle in these animals, and in just gross morphology, you can see control are on the left and shunts are on the right, and the heart picture is shown. And the RV is clearly hypertrophied, and then when you look at the kind of cut section, you see that there's really biventricular hypertrophy. And particularly if you look at the free wall of the RV and the shunt in animals, it clearly is more hypertrophy when compared to control specimens. We then challenged these animals with an acute PA banding protocol where we increased the RV pressure to about half systemic pressure, and we maintained that level for 4 hours. And what we found was that shunt animals had increased cardiac output than did control animals, despite the increased afterload, and that although control animals were able to maintain their baseline cardiac output, they really did it by becoming progressively tachycardic, which you can see in the bottom left panel, whereas shunted animals actually increased their contractility in response to this increase in afterload. And this kind of second slow increase in contractility is in the left ventricle what's described as the ANREP effect. It's basically how we maintain cardiac output in the face of increased afterload in the LV in a sustained manner. Classically, the RV has not been shown to demonstrate this effect, but this increase in contractility represents this kind of novel finding of an RV ANREP effect. And this was despite the fact that the shunted animals had lower increase in their serum catecholamines than did the control animals. And so that may explain some of the different tachycardic response that we saw in the controls, but both epi and norepi levels were lower in shunted animals. And finally, kind of what the job of the right ventricle to do is to pump to the system that it's coupled to. And so in pressure volume loop studies, that's termed ventricular vascular coupling, or the ratio of the slope of the ESPVR relationship, or EES, to EA, which is vascular impedance. And that relationship was preserved in shunt animals, but was uncoupled in control animals. So basically, the shunts were able to increase their contractility in parallel to the increase in afterload, whereas the controls, that relationship fell apart. And that's often a marker used to follow patients with pulmonary hypertension. There was also evidence that the right ventricle in the shunted animals was more efficient than was that of controls. And so, as I said in one of the early slides, the stroke work done can be represented by the total area, or what's shown in blue in that top graph, of the pressure volume loop. And that if you include the line drawn out to V0, that's the potential energy. So if you think kind of back to physics, so total work done, and then the external work is really the stroke work. So the ratio of those two is kind of a ratio of the efficiency of the ventricle. So pressure volume area was increased significantly in the controls after PA banding, but not in the shunts. And that also correlates with myocardial oxygen consumption. Whereas efficiency, again, the ratio of sort of external work to total work was preserved in shunts, but was not preserved in control animals. We then began to get into some of the mechanism of why this might be. Without going through too many of the details, basically nitric oxide signaling is preserved at fetal levels in all isoforms of nitric oxide, both at the RNA, and I'm not showing it, also at the protein levels in these animals, in the RV, but not the LV. And then finally, when you look at nitric oxide activity, it's also higher among the RV of these shunted animals, and that's similar again to fetal levels. And this may provide some mechanism for the observation that we had of the increased ANREP effect in the shunt RV, in that eNOS was more associated within the cardiomyocytes as opposed to simply at the endothelium in the shunted animals. And eNOS phosphorylates nitrosylates the ryanodine type 2 receptor, and that leads it to be more in the open conformation and allows more calcium efflux. And so that provides then a mechanism linking increased nitric oxide expression and activity, its localization within the cytosol, increased nitrosylation of this receptor, which we demonstrated is a purported mechanism linking back to actually increased function with contractility. So there we spent quite a while talking about preserved systolic function that we've observed in our congenital heart disease model in the face of increased afterload, and now looking at preserved ventricular interactions. And so one of the ways that we challenge our animals in the lab acutely is with a thromboxane A2 mimic, which acutely raises pulmonary vascular resistance. The molecule is called U46619, if you're curious. And what we find when we add that, and actually if you start just by looking at the shunt tracing at bottom, this is kind of the pressure volume trace broken out over time with pressure and volume given their own lines. That's kind of the typical pattern, the shunt at bottom, where you see a rapid rise in LV volume in diastole and kind of a smooth decrease in that with ejection. What we find when we increase PVR in the control animals, but not the shunts, so going back up top to this red circle, is that it develops this kind of biphasic increase in LV volume. And so it seems to be dependent on atrial kick for full filling, indicating diastolic dysfunction in the LV, not the RV, of these animals. And so that would be an indication, again, that as we know, as the RV begins to fail, it impinges into the left ventricle and affects particularly early diastolic filling of the left ventricle. So evidence of adverse ventricular interactions, just looking at the trace, but then when we quantify that using LV stiffness in the setting of this PVR increase, we find that shunt animals have preserved or actually slightly lower than baseline levels of LV stiffness, but both control and another model we have LPA ligation animals, it goes up. Right ventricular contractility, as measured by ESPVR, is augmented, where it's only slightly augmented among control animals. And again, that ventricular vascular coupling measure, ES to EA ratio, is preserved in shunts, but not in controls. And so evidence of beneficial ventricular interactions, or at least preservation of baseline in the setting of an increased afterload. So next, looking at angiogenesis, and this again gets back to the point I made at the start of the talk, that many models and the way we even talk about patients talk about a period of compensated hypertrophy that, again, inexorably declines to RV failure. But I think that there may be a different kind of paradigm with this that we could think about, where perhaps if we have a adaptive remodeling phase that we could preserve, that may have fundamentally different pathways that are involved than the kind of typical pathologic spectrum, that may be a tool we can harness as a therapeutic. So this is obviously not my schema, but part of the hallmark of this phase of compensative or adaptive remodeling, as they term it, is that the capillary to myocyte ratio is preserved even in the face of hypertrophy. And you can imagine, as that ratio goes away, and you can see in their cartoon of maladaptive remodeling, where there are fewer capillaries despite ongoing myocyte hypertrophy, that myocardium then becomes energy starved, especially in the face of increasing amounts of work that's expected of it. And so one of the hallmarks then of adaptive hypertrophy is not just the physiologic performance that I've shown you, but what their angiogenesis and capillary ratios are. We found that the capillary density in our shunted animals in the RV, this is staining where CD31 is shown in green, which is an endothelial cell marker. DAPI is a nuclear marker, and then it's really not showing up very well there, but TNT is a red or myocardial myocyte marker. And so we see increased endothelium and increased capillary density by the CD31 ratio in our shunted animals, which is important because, as I showed you early on, they were more hypertrophied in the RV compared to controls. And one mechanism that might explain that is VEGF, which is many of you know is a pro-angiogenic protein, is more highly expressed in shunted animals, and again, similar to what's there in fetal life. And then lastly, lack of fibrosis despite RV hypertrophy would be another hallmark of an adaptive RV. And we focused on this microRNA 29A. In part, it's interesting, it's actually very highly expressed among physiologic hypertrophy, such as swimmers or runners, both in animal models and in humans, who have obviously adaptive hypertrophy. And so we looked at, in our model, and the gray bars are shunted animals here, and microRNA 29 was much more highly expressed. And that correlated with downstream targets of microRNA, which microRNA inhibits downstream. Both collagen 1 and collagen 3 were decreased, and fibrosis was decreased slightly in the shunted RV compared to the control. And so again, just kind of driving home this point that we believe that these are global changes that are present, that while we are kind of, the work I've done so far has been going after specific pathways and trying to understand our initial sort of exciting physiologic findings, that there may be a more elegant way to do this and a more comprehensive way to do this. And so we've started doing more transcriptomics and computational work in order to get at changes that may not be the kind of classical pulmonary vascular changes or pulmonary cardiac hypertrophy changes. And so this is what happens when you pair our data set from RNA sequencing with our microRNA data set. And obviously, I'm sure you can read all of that perfectly. But just to show that some of these molecules, especially microRNAs, are really pleiotropic and have multiple downstream effects. And beginning to think about it in these kind of transcriptional pathways is something I'm frankly just really learning how to do more and more. But I think that may be a way to ultimately change this paradigm rather than kind of going after targets that have already well worn. And we already have found some kind of novel pathways in our analysis that point in new directions. And one that I think is particularly interesting is that telomere maintenance is preserved or increased among our shunted RV compared to control RV. Not quite sure what that means yet, but I think that's the kind of novel direction that this type of work can take us in. So for the last couple minutes, I just want to go in a little bit of a different direction and sort of show what we can do within the Pediatric Heart Center, and I think a nice example of some collaboration between the disciplines in the Heart Center. When you Google UCSF collaboration, this comes up. These are some radiology residents who are collaborating at the interactive board in the lobby. And so the work we've done is a group effort between what I'll show you, a company actually, a startup that makes this molecule Omniox, Emma Maltepe, who's a neonatologist here, Jason Boehm, who's in our division, but this first paper is his work, and then the final studies I'll show you are some bypass studies that we've done with Dr. Reddy helping us with the kind of conception, and then with Neil and Clint helping us with the execution and actually coming to the lab and helping us put some lambs on bypass. And so this OMX molecule is an oxygen-carrying molecule, and I won't go into all the chemistry part because I cannot, but it basically delivers oxygen at very low oxygen tension, so something like 10 torr. So it works very well in extremely hypoxic environments, and that also avoids some of the earlier kind of oxygen replacement or hemoglobin replacement molecules were found to be quite toxic because they actually delivered too much oxygen and caused oxidative stress. So this kind of avoids that problem. And so a paper that's in press now, we did an experiment where we subjected lambs to hypoxia, so 10% FiO2. And we found that in animals given this molecule, at 1 hour they had preserved systolic function of both their RV and their LV. And you can see example loops here. So the baseline for controls is in black, and then after 1 hour in green, you can see their function suffers quite a bit. Whereas in the animals given Omniox at 1 hour, their function is preserved. It looks like it's increased, but it actually statistically is not above baseline. And as I said, that was true for both the RV and the LV. And again, this increase cannot be explained by more catecholamines among this group of animals. So that's just kind of a background study. What we've done and have data I'll show you here is that Dr. Reddy and Neil came up with us with a protocol for doing bypass and causing some myocardial injury that was reproducible. So what we did was put animals on bypass, but before delivering the cardioplegia and going cross clamp, use coronary occluding loops and occluded about the distal third of the LAD territory, where the homonymous artery is called in sheep, to have a part of the LV that would not be protected by the cardioplegia. We then went on cross clamp, delivered a couple doses of cardioplegia that obviously did not go to the protected area, and then before the final dose, removed the vessel occluding snares and gave cardioplegia that either contained vehicle or this OMX molecule, so it was only in the cardioplegia solution, and then rewarmed and measured myocardial performance. What we found was that after 3 hours from separating from bypass, that control animals, and you can see, to make it confusing, I changed the colors. Blue is in baseline for controls, and then 3 hours off is in black. You can see they suffer decreased systolic performance, whereas the OMX animals, again, where red is baseline and purple is after being off, really had no change in their function. And that was true. This is just 3 and 3 right now, so still relatively preliminary. But that was still true when we looked at their contractility. They really recovered from the initial insults that both groups suffered at 30 minutes off bypass. By 1 hour off, they were back to baseline, and that was sustained throughout 3 hours. And people who've been there in the lab know that 3 hours at the end of a bypass day is quite a long day, so we were happy to end there. And then cardiac output was also at 1 hour preserved in the OMX animals, but not in the control animals. So again, preliminary data, but I think an exciting example of what this group can do together, but also what kind of a plug for our lab as well, that really is a preclinical model, as a large animal model of ways to test hypotheses that are clinically relevant. This drug is already being used in humans in the cancer world, and so may have promising applications in our world as well, we hope. And with that, I'll say thank you for your attention, and thanks for not being alarmed by my hyperventilation. And thanks to everyone in the lab, especially Jeff, who's out of town today, but is a really wonderful mentor. And again, to Neil and Clint, who came and helped. Thanks very much.
All right. I also want to thank the PHC Research Committee for inviting me as part of the group to talk today and present our work, ongoing work on the pulmonary lymphatics in the model that Rebecca described. I don't have any excuse for being short of breath, so that's my only disclosure. So just by way of introduction and kind of setting the clinical question, patients with respiratory dysfunction are known to have, or sorry, patients with congenital heart disease are known to have significant respiratory distress, and the respiratory dysfunction associated with their lesions can really drive a lot of comorbidities. And I think we've seen that in patients with common defects like VSD, and also with patients before repair with truncus arteriosus or transposition, where their altered mechanics really drive respiratory distress, poor feeding, and poor growth. And it's been known for many years that this dysfunction, well, it's been studied for many years, actually since the late 60s, in a number of settings with patients with congenital heart disease and evaluation of their respiratory mechanics. And all these studies that I'm kind of popping through have consistently shown that there's a decrease in compliance, in lung compliance, before surgery and also after surgery. In this collection of patients analyzed, adults with congenital heart disease, several years out from their surgery, the degree of pulmonary dysfunction or respiratory mechanical dysfunction, moderate to moderately severe dysfunction, correlated with decreased survival. In these patients, the degree of dysfunction is also associated with the complexity of the lesion, which makes sense, but it also serves as an independent predictor of mortality. And what's really important, I think, is that these abnormal respiratory mechanics that I'm describing, either altered airway conductance or increased recoil resistance or decreased lung compliance, these abnormal respiratory mechanics don't necessarily improve completely after surgical repair, even after a successful surgical repair. So in these two related studies out of the University of Prague, the first published in the late 80s and the second follow-up study in the mid-90s, the first looking at simple transposition where they were corrected with mustards or Sennings, and then VSDs closed with patches. All of these studies happened, all these evaluations of respiratory mechanics occurred between 4 to 9 years on average after their successful surgery. So not right after, not post-op, not with chest tubes. These are patients who are living their lives out there. And they all showed, a large majority of them, 88% in the transpositions and 70% in the VSDs, significant alterations or dysfunction in their respiratory mechanics, particularly with lung compliance. What's also been known for a long time, for almost the same amount of time, is that patients with congenital heart disease, particularly with increased pulmonary blood flow, also have altered pulmonary lymphatics. This micrograph from Dr. Stanger, which he took himself in the late 60s, is an autopsy specimen from a child who died with an unrepaired VSD. And it also shows some really impressive dilated pulmonary lymphatics. Of course, other cardiac lesions are also associated with lymphatic dysfunction and alterations in the lymphatic architecture. Patients with hypoplastic left heart syndrome with a restrictive atrial septum have these classically dilated lymphatics stained in brown, compared with normal control patients, and also with hypoplastic left heart patients who have inadequate atrial communication. So this dilation is well known. Also in patients with obstructive pulmonary veins. So for example, in this model that was developed, a large animal model of obstructed veins developed by Peter Oishi and Gary Raff several years ago, we were also able to identify these really massively dilated lymphatics. Here staining in yellow, which is double stained for CCL21 and LIVE1, which are two lymphatic specific markers. So just as a step back and as a reminder, the lymphatic vasculature is a completely separate vascular system, separate from the arteries and veins, and it really is everywhere in the body, and incredibly important not only for fluid homeostasis, but also for immune surveillance and immune function, lipid metabolism, and in many cases it's much better recognized as a mechanism for metastasis with a number of cancers. I just want to point out, which is nothing I'm going to talk about today, but for the longest time, people felt like the CNS was kind of a preserved space and lacked any lymphatic architecture whatsoever. And just in the last 3 years, there's been a lot of advance in identification of G lymphatics and also true lymphatics. So it's now becoming much more clear that there's actually lymphatics even in the CNS, probably responsible for CSF clearance. And it's something that we can touch back on later on at the end of the talk, what I think is actually going to be quite interesting. But for CSF clearance and as well, people think for importance for washing the brain while sleeping. So another reason not to take overnight call. So although it is a separate vascular bed, the lymphatics, particularly the initial lymphatics, are really quite intimately connected or intertwined with the capillaries and the venules. And this relationship, this intimate relationship, is really important to allow solutes and interstitial fluid that accumulates here in this interstitial space to clear through the initial lymphatics and then get transmitted down the muscularized collecting vessels and then back into the venous system. And so for the last several years, it's this relationship that we've been really interested in, the normal biology of the pulmonary lymphatics, but also the aberrant function and development in the context of increased pulmonary blood flow and congenital heart disease, because the increase in flow and the increase in pressure is transmitted all the way through to elevated capillary pressures, which then drives, through Starling forces, increased accumulation of interstitial fluid. And we think, and we believe that this increased accumulation of interstitial fluid really drives kind of altered mechanical forces, flow-induced altered mechanical forces for a prolonged period of time, and that results in dysfunction of these lymphatics. And that's what we've been trying to assess. And I think this might really contribute a lot to the respiratory dysfunction that we've identified in these patients with congenital heart disease, even years after their repair. So it's something that I think we might be able to target either in the early convalescent stage, certainly in the preoperative stage, but that's really the purpose or the point of studying these vessels in this vascular bed. So to do this, we use the model that Rebecca just described in great detail, and I think it really highlights the importance of the strength of the model to look at cardiopulmonary interactions. And so we study these animals at about 4 to 6 weeks of age, at which point clinically and radiographically they look just like the infants in this step-down unit or in the ICU who are awaiting closure for their VSD. They're tachypnic, they don't eat well, and they don't grow well. Radiographically, either by chest x-ray or by CT scan, looking at controls on the left or shunts on the right, they have this massive cardiomegaly, which Rebecca really explained in great detail. They also have these arterial and venous distension. These are the pulmonary arteries in normal animals and in shunts. There's interstitial edema marked by these red arrows. And actually, I think more impressively, what we've also found is that in these animals with increased pulmonary blood flow, they have a massively disrupted or distorted postnatal pulmonary architecture, pulmonary lymphatic architecture. So these are shunt lambs at 3 weeks of age compared to their control siblings. And so I think we started to really get at the underlying mechanisms that drive this developmental change, but I think if there's time at the end of the talk, I can touch on some of that, and Rebecca alluded to those pathways as well in her talk, but I think I really want to spend most of the talk really talking about in more detail the functional disruption that we've learned about in these animals. And so the collecting vessels, lymphatic vessels like the thoracic duct are worked before. These vessels are muscularized and they pump with their own intrinsic mechanism, and I think this pumping is essential for their function. A lot of labs are studying about how this happens, but some of it's flow-mediated. What is known and what's been shown by a lot of labs is that nitric oxide and endothelial nitric oxide signaling is essential for this normal pumping and normal flow. And so just a little bit of evidence for that is shown here. This is a pressurized canine thoracic duct system. So they've taken thoracic duct from dogs, mounted them on cannulas, and pressurized the system and flowed through there. And if you block nitric oxide synthase, and so therefore block the ability to make nitric oxide, you can drop the flow-mediated relaxation that's seen. And this is from Ohashi's group in Japan. Similarly, using a genetic model, using an in vivo mouse tail model, if you pharmacologically or genetically inhibit eNOS, which is endothelial nitric oxide synthase, which is the nitric oxide synthase in the endothelium that makes nitric oxide, if you block either of those, you can drop lymph flow velocity significantly. So it's clear that nitric oxide signaling seems to be important for lymph flow. So with that in mind, we hypothesized that chronic exposure to increased pulmonary blood flow in our model of congenital heart disease should injure the lymphatic endothelium, disrupt nitric oxide signaling, and thus impair lymph flow. And the way we went about trying to test this hypothesis was kind of a three-pronged approach. The first was direct cannulation of pulmonary lymph in the intact animal. The second would be harvesting of the thoracic duct, slicing them up into rings, and then placing them on wire myographs to quantify pressure diameter relationships, or pressure tension diameter relationships of those rings themselves. And the third would be isolating and culturing lymphatic endothelial cells, and then really getting at the biochemistry and the mechanisms, the pathways that drive this nitric oxide dysfunction. The first approach really goes back to some pioneering work done by Norman Staub's group here at the CVRI and UCSF in the 1960s and 70s, and then extended in Michael Mathey's lab through the 80s. And this really involves direct cannulation of the efferent vessel of the caudal mediastinal node, schematized here in green, and the head of which you can see here that's dyed blue. This caudal mediastinal node is this long sausage-shaped lymph node that drains the vast majority of pulmonary lymph in normal lambs. It usually has one efferent vessel that tracks off of it and then eventually dumps into the thoracic duct, which is right here. And so what these labs did several decades ago, and what we did, is cannulate this vessel to directly collect and analyze the lymph from the lungs, essentially, of control lambs and lambs with these shunts. We also took thoracic duct vessels, harvested thoracic duct vessels, cut them into rings, mounted them on these pins, where we could then quantify the relationship, the tension basically that those rings could generate under different conditions, either normal baseline conditions or under conditions of exposure to vasoactive medications like norepinephrine. And the results of these kinds of experiments are summarized here. First, I'd just like to say, though, that using the CT lymphogram, we were able to first show that by tracking the dye across, in serial scans over 15 minutes to 75 minutes, we were able to show that in shunt lambs, transit time across the pulmonary lymphatic bed, so from beginning through to the thoracic duct, was 5 times slower than in controls. You could track hospital units and intensity and basically show that there was a real delay in the clearance of dye across that pulmonary bed. So there was a real slowdown in the transit of fluid across. And this correlated with a decrease in bioavailable nitric oxide that we measured directly in the lymph that we collected from the control or shunt lambs. So in blue here, this is shunt, and this is orange control. This is bioavailable nitric oxide, or the active metabolites of nitric oxide, and that's decreased significantly in shunts. When we looked at the rings from from either shunt or controls, the thoracic duct rings, this is just looking at the baseline tone. And if you just concentrate on the y-axis here, this force scale here is 200 milligrams, and in the shunts, it's 600 milligrams. They have a much higher baseline tone. And in response to increasing doses of norepinephrine, they have a much more robust contractile response as well. They're much more muscular when you look at them by histology. In addition, another really interesting point here is that when they were given acetylcholine, which is a typical cath lab test to try and test for endothelial function, normally what would happen is the endothelium would respond to acetylcholine and then release nitric oxide that they produce, allowing the smooth muscle cells that surround them to relax. So the control rings, will relax with increasing doses of acetylcholine. As you can see, the tension drops nicely, but in shunt rings, the tension remains the same. They're not able to relax. However, if you give them exogenous nitric oxide with an NO donor like SNAP, both control rings and shunt rings can relax appropriately. So this suggests that there is some endothelium-dependent, some attenuation of endothelium-dependent relaxation, or some disruption of NO signaling. All this together suggests that the NO signaling is not quite right. So to get at the kind of biochemical pathways that might drive this, we, again, isolated the endothelial cells from this efferent vessel, which in vivo was seeing either normal flow of lymph in the control animals or much higher lymph flow in the shunts. So we know that when we measured it and analyzed it, we were getting double the flow in shunt lambs compared to control, something like 10 microliters per liter per minute versus 5 microliters per liter per minute. And this is for 4 to 6 weeks before we actually harvested these cells and grew them in culture. And what's interesting is that even in culture, after they were no longer exposed to the same mechanical forces, they still maintained the phenotype, which is summarized in 2 slides from now. But I just want to show you that these cells we isolated, and we're able to separate them from their smooth muscle cells and show that they were pure populations of lymphatic endothelial cells. And we did a number of experiments, which I don't want to go into right now, but just to summarize that we basically identified 2 pathways at least that contribute to the decrease in bioavailable NO that we measured directly in the lymph. But the pathway essentially starts with increased pulmonary blood flow associated with that large vascular graft and unrestricted left-right shunt. That drives increased capillary pressure, which we also measured essentially as double normal, so 15 millimeters of mercury versus 7 in the animals that we had tested, and also driving double the pulmonary lymph flow. One pathway that we studied earlier and that Rebecca also mentioned in her talk was the NOS pathway, or the nitric oxide synthase pathway, which produces nitric oxide. In our system, under these conditions, in the lymphatic endothelial cells, two things happened. One was that eNOS, or endothelial nitric oxide synthase, was downregulated both in expression and activity. And then iNOS, or inducible nitric oxide synthase, which is involved in inflammatory processes and is very efficient at making reactive oxygen species, was upregulated, again, both mRNA and protein expression as well in activity. And so together, this downregulation of eNOS, upregulation of iNOS, drove production of reactive oxygen species, which was scavenged by available nitric oxide. In addition, we investigated the PPAR gamma signaling pathway, which was worked on in large part by a postgraduate who actually is starting med school this month, and she just published this earlier this year. But essentially, KLF2, which is a flow-induced transcription factor, it's a transcription factor that's produced when the cell sees or is exposed to flow-induced shear forces, is upregulated, which then directly downregulates the expression and activity of PPAR gamma. Normally, PPAR gamma suppresses NADPH oxidase activity, and in this case, when it's downregulated, NADPH oxidase activity goes up, and again drives reactive oxygen species to accumulate. So both of these pathways at least contribute to the decrease in bioavailable NO that we see, and thus the impaired pulmonary lymphatic function. So that doesn't sound very good, and I think then we kind of asked ourselves the same question that I think every one of you have asked me when I've been on call or on service, which is, what do you do about it? And give me something, give me something. So we asked whether or not we could try to boost bioavailable nitric oxide and rescue or modulate pulmonary lymphatic pumping. And so to do this, we did the following experiment. We laid out control and shunt lambs and directly cannulated that efferent vessel to collect the lymph so we could analyze it and monitor flow rates. And after we got a baseline set of measurements, we added inhaled nitric oxide at 40 parts per million and then measured before and after the fact the levels of bioavailable nitric oxide and cyclic GMP. And what we saw was that very quickly after starting NO therapy, we saw an upregulation of both bioavailable nitric oxide and cyclic GMP. I just want to point this out because it might be confusing, but this is the change in NO from baseline, which is why the shunt seems like it's higher than the control, but it's starting from half the absolute value, and so it increases significantly more because of the exogenous delivery. But both increase significantly, nitric oxide and cyclic GMP in the controls and the shunts, and certainly by 45 minutes they're saturated with both. As you would expect with nitric oxide, there's a decrease in pulmonary artery pressure in both the controls and the shunts and the calculated pulmonary vascular resistance, and it's about 15% in each. As I told you earlier in the talk, the real driving force for interstitial fluid accumulation is Starling forces, or hydrostatic forces driven by the pulmonary capillary or pulmonary artery pressure. So you might expect that lymph flow would decrease, the absolute lymph flow would decrease when the PA pressure fell. But in fact, what we saw was there was a preservation of the rate of lymph flow. So there was no drop in the absolute quantity of lymph that we're collecting, even after starting nitric oxide. And in fact, if you normalize for the pulmonary vascular resistance, we saw a relative increase in both controls and shunts across the time period that we studied them. So just to summarize, I think I'm running out of time here, but just to summarize this part of the talk, I think nitric oxide signaling we've shown pretty well is disrupted in the pulmonary lymphatics of these lambs that have chronically increased pulmonary blood flow. And I hope that you feel like there is some potential for therapies targeting some of the pathways that I've talked about, either PPAR gamma signaling with a PPAR gamma agonist, or nitric oxide signaling with NO donors like sildenafil or tadalafil. And certainly inhibitors of NADPH oxidase I think are also quite promising as well. So all of these we need to study in vivo. We're in the process of doing that with our lambs, either controls or shunts. But I also think there's some value to using the wire myographs and the pressurized myographs, which we have access to try and study really what these are doing at the functional level to lymphangions as far as their pumping, the contractility, the amplitude and frequency of their pumping, and also the flow through that functional vessel. And so I just wanted to give you a little taste of what some of the experiments that we've been doing and what I think are questions that were worth asking. Just one of the things we also talk about often is what are diuretics doing in these patients. We know all of them are on diuretics. Oftentimes, even after surgery, they go home to convalesce with a package of diuretic cocktails. But we don't really know what that's doing to the lymphatics. I think there's evidence that there's a venous dilatation that's been postulated as a mechanism for clearing lung water, even in patients who don't have kidneys. But I just wanted to show you this example experiment of one control animal where we took thoracic duct rings. These are 8 different rings from that animal on different channels. And you're just seeing the kind of measurement that we can make, which is, and you see there kind of automatic pulsatility there. We can measure, for example, the mean tension over time. We can measure the frequency of pulsation, and we can also measure min-max or the pulse pressure differential. And in this case, in response to increasing doses of acetazolamide. So here we've asked acetazolamide, we've tried furosemide as well, and this graph just summarizes the first set of experiments that we've done for that. Again, Lasix is on the left and Diamox is on the right. The frequency of pumping is in blue. The mean force or the tension that those rings are generating is in green, and then the pulse pressure or the min-max amplitude is in red. And both diuretics, both furosemide and acetazolamide, seem to decrease the mean tension over time about 20% across the dosing curve. And as well, they slow down the rate of pumping, which is interesting because what we're trying to do, I think, what the general feeling is that when you turn that vessel into a big, wide tube, you can flow things more easily through there, which makes sense. What I think is quite interesting is that with acetazolamide, more than with Lasix, it looks like that pulse pressure is actually increased about 20%, which could suggest that there might be augmented pumping or augmented contractility, which you can't really tell with a ring because it's just a cut piece of vessel. But what we need to do is take a full functional lymphangion, mount them on the pressurized myograph, and then we can really get a sense of what acetazolamide or anything else is doing to the flow and the contractility. But what would be great is if we can really show that we can augment pumping and augment fluid clearance in these ex vivo models and also obviously in the in vivo setting. So hopefully there might be some therapeutics to augment lymphatic function and to help support the patients as they recover from congenital heart disease. Obviously, all of these drugs, I guess I should have said, the caveat to all of them is that before they're repaired, any of the drugs that dilate, any of the drugs that, like sildenafil, for example, is a risky maneuver because you can potentially increase pulmonary blood flow even more and flood the lungs if the left ventricle can't handle it. But I think after repair, certainly during the convalescent phase, we can certainly make a change in the way these patients breathe and recover at home. So with that, I'll stop. And I just want to acknowledge the large group of people who do this work, starting with Jeff, who many years ago is the one who came up with this project to study and really kind of pushed me down this path and has been incredibly generous with his time and unwavering in his support. I really want to highlight that Rebecca, Jason, and Peter in the lab, and then as well the great collaboration with Emin and all the people up at Davis, including Gary Raff and Linda Talkin. So with that, I'll take questions. And this is my funding.
I think we have a few moments left for questions for either Sanjeev or Rebecca. Does anyone have any questions at all? How about online?
Sanjeev, any sense for what the timing effect is around this? It seems to be how much of this is acute and chronic. You were implying that many of these patients have effects decades after due to this, and a lot of the intervention you're talking about is acute effect on acute pumping.
Here.
So, yeah. So I guess my question is, how much of this is sort of an acute dynamic balance, and how much of it is a chronic abnormality of the fluid balance in the lung that impacts the compliance? Any sense for that?
Yeah.
Sanjeev, can you repeat the question for the remote, please?
Sure. Dr. Moore was asking how much of this effect might be related to acute changes. Let me see if I got this right, but acute changes versus the chronicity that we're talking about. The model is a chronic model. The acute dosing of the drugs would not be, at least in these experiments, won't reflect that chronicity. So I think, two things. One, I would say that we've actually, in the initial experimental setup, we actually compared this model, which is the fetal placement of the shunt, and then studying them out 4 to 6 weeks after they were born, versus an acutely placed shunt at 4 to 6 weeks. And in that case, when that shunt was placed, lymph flow went up, PA pressure went up, lymph flow went up proportionally, and in fact, lymph flow went up in a way that what we were able to determine was that, that it looked like it was an adaptive mechanism, as if you were exercising. So normally when you exercise, your flow goes up, your pressure goes up, and you don't flood your lungs and you don't get lymphatic dysfunction, I don't think. So there was accommodation, essentially. So in that respect, I think we were able to tease apart chronic versus acute. Now, the drug treatment we need to give chronically and then look at lymphograms and then look at lymph flow at 4 to 6 weeks of age. We still have to do that. And I think that'll get at the question that I think you're asking, which is how do we see the effect of, or the benefit of those drugs? What I would like to do is give, say, for example, either sildenafil and or sildenafil and diuretic before the shunt's closed, and then close the shunt, and then test it again after the shunt's closed. But in order to do that, we need to plug it or snare it, which we haven't. We've done closure in the acute setting in some of Rebecca's studies, but we haven't done that chronically, at least for my studies yet.
Sanjeev, I have a question because your model is really what we see in the preoperative period, and we struggle with a lot of this postoperative. We kind of sometimes say that it's due to increased right-side filling pressures is where we tend to see with kids that have more pulmonary dysfunction and more lymphatic drainage.
Mm-hmm.
I assume that has something to do with that initial model you showed about how the lymphatic system is eventually going to drain into the venous system. But do you have any information specifically about patients where there isn't an increased shunt, but they're still having a lot of problems with the lymphatics? And whether it's due to increased filling pressures or whether that's just something we've made up?
No. I think so. Two things. I think those patients like Glenn's or Fontane's, I think they've had multiple hits. I think early on they may have had increased pulmonary blood flow, so their lymphatics were maybe already primed with a first hit, and then either whatever the changes in afterload or filling pressures are driving the chronic leakage. That might be one mechanism, but having had that first hit. But I think the pressurized myograph system is a great way to study that because I can actually adjust the afterload in real time and then see what the effect is, not only on the pumping, on the amplitude, on the frequency, but also on the contractility. And I think that will get at some of these questions because, let's say, I mean, one thing I would say is that if I blew through the hemodynamics pretty quickly, but the CVP was not any different in the shunts and the controls. So the afterload for the thoracic duct was the same. But I think it's worth modifying.
I think that's more of a postoperative.
Yeah, no, for sure. I think so. Similarly, I think we need to close the shunt and then see how those animals respond because the lymphatics don't recover immediately, if at all ever. We don't know, actually. So.
And the trisomy seem to be worse. Do you have trisomy 21 sheep?
We're gonna make some CRISPR-Cas and get right on it.
Great. Thank you, everyone. Please join me in thanking Sanjeev and Rebecca. Okay.